Okay, so 7-3 is about cell transport, okay? Cell transport is the transportation of materials in cells, okay? There's two types of cell transport. There's passive transport and then there's active transport. First type, which is called passive transport, means that it does not require energy. It does not require energy. So passive transport does not require energy because the material is moving from high concentration to low concentration. Okay, that's why it does not require energy, because you're going from a high concentration to a low concentration. This is called, and she made us write this quote down or with its gradient. Its gradient is talking about the shift between areas of different concentrations, just like an ombre or gradient effect where it goes from like one color to the other color. Yeah. Okay, so anytime, like, also if it's like, oh, it's going with the grain, stuff like that. It always moves from high concentration to low concentration. And I'll explain that later. If you're confused, just let me explain that later. Think about this. You are on a roller coaster and you're at the top of a roller coaster. When you're going down that huge hill thingy, do you need added energy? No, you don't. Because you don't need added energy. This is passive transport. There's 3 types of passive transport. There's osmosis, which is the movement of water from areas of high concentration to low concentration, okay? For example, right here up top, we have something where it's like we have 2 H2O molecules, and then we have, for example, we have 4 H2O molecules, and then we have 2 H2O molecules on the outside. So movement of water from high areas of concentration to low areas of concentration means that this will give 1 molecule of water to this area, because the goal of passive transport, all these transports, is to reach what is called as equilibrium, okay? I'll explain equilibrium later. Just osmosis is the movement of water from areas of high concentration to low concentration. And sometimes osmosis can be facilitated diffusion, which is another type of passive transport. Facilitated diffusion is where it needs help in order to move from areas of high to low. Okay, for an example, if you watch my lecture notes on phospholipid bilayer, how I told you that the small polar molecules go through integral proteins, they need facilitation, they need that help. This is facilitated diffusion. It needs help. And osmosis can sometimes be a type of facilitated diffusion, because if you watch my lecture notes, you would know aquaporin helps water move. So that's a facilitated diffusion. The third type is simple diffusion, which is just the movement of everything except water from areas of high to low concentration without help. So simple means that they don't need help, they're good, and it's everything except water. This is like really simple. She'll probably give us like examples, but it'll probably be straightforward, not gonna lie. I don't know, to be honest. Second type is active transport. Active transport is transport that needs additional energy. Okay, it needs energy. There's 2 reasons why this needs energy. Number 1, because it is going up or against its gradient. For example, we have an area of a low concentration and we're going to an area of high concentration. This is an example of active transport because you are going against the gradient, because it always goes from high to low. So when you're going from low to high, you need to add energy to let it go from low to high, okay? And then another reason why active transport needs energy is because of like large amount of materials or large sizes of materials. And let me explain. So there's 2 types of this, okay? There's exocytosis, which means it goes out of the cell, and then endocytosis, which means it enters the cell. Let me show you. So right here, we have, this is like materials, okay? So, and we learned this, this is a protein inside a vesicle, and then it goes through the Golgi apparatus, it gets, changed through the Golgi, and then it comes here to the edge of the cell. And it's saying, I want to go through the plasma membrane, like, let me go. But for it to go, it needs energy. It needs additional energy. It can't just go through. It needs that additional energy. This is exocytosis because it's exiting the cell. Endocytosis, on the other hand, is where, basically, so we have, some materials, for example, okay? Materials out here. The materials want to go inside. They want to enter the cell. How are they going to enter the cell? Well, the materials, like this, it's going to come to the edge. These sides of the plasma membrane are going to pinch together, and it's going to create this kind of like pocket structure. And this is going to go inside the pocket. And then it's going to all close together, and then it's going to create its own materials inside a vesicle membrane. That's basically what endocytosis is. It's just the like enfolding or like kind of pinching of it. I think that's how the book explained it. Hold up one second, please. It is exocytosis. Yeah, it's basically the material of cell by infolding or like the creation of pockets like this. There's two different types of endocytosis. There's phagocytosis, which, phago means like food or solids. Phago means solids. The way I remember this is phagocytosis. Like if your friend is being annoying and they're like eating all your food during lunch, you would call them like a faggot or something. So phagocytosis is with foods or solids, like that's like foods or solids are moving. And then pinocytosis is liquids. She didn't really go into a lot of detail because we're gonna learn this like later on in the year, I think. But I would recommend reading the book on this lesson. A little bit of phagocytosis is that it's a type of endocytosis. The food, like it's a food vacuole, so like food material will go inside. Just like how I told you, the endocytosis creates the pocket. It's gonna create like a vacuole. And then white blood cells. Just read the book on this part. And then there's equilibrium, okay? So for equilibrium is where there's no gradient, okay? When I mean by gradient, I mean that there's like areas of high concentration and then there's areas of low concentration. There's no gradient. It means that it's equal on each side. The concentration is equal on each side. Cells or like diffusion will always occur until equilibrium is reached. Equilibrium here. We have 2 H2O here and 4 H2O here. It's going to give 1 H2O, 3 H2O, 3 H2O. That's equilibrium. And a good example that Miss Romana gave is if there are 27 students in a classroom and then there's exactly 27 students waiting outside the classroom, when those students switch with the students inside the classroom, that's equilibrium because it's exiting the same that it's entering. There's no gradient. There's no differences. That's cell transport. Tonicity, it sounds confusing. And Afra told me that even up until her DAT or dental admissions exam, she still needs to know this. So you really have to pay attention and also watch videos like this video and other videos can help you. Tonicity. Tonicity refers to the concentration of solutes in a solution. It basically compares the concentration of what's inside something versus what's outside of something. Hold on with me. There's 3 types, okay? 3 types right here. Number 1, hypertonic solutions. Hyper, hypertonic, okay? This is an example of a cup with some water, and this is a cell, okay? Inside this cell, we see a low concentration of a solute, and we see a high concentration of water inside the cell. When it says a high concentration of water, we understand that this cell has a lot of room to move around. Like inside, the solutes have a lot of room to move around. They're like spaced apart. Whereas, basically, so the outside, so this is low concentration of solute and high concentration of H2O. Whereas the water has for an example, this is a hypertonic solution. The water has a high concentration of solutes and a low concentration of water, okay? So, water always goes from high concentration to low concentration. Always. So, this, because there's a lot of water in here, water is going to start to exit. If you might be asking, why is it water that's exiting and not the solute that's exiting? Because you're saying it goes from high to low. Well, the cell membrane, and I want you to really write this down if it helps you remember. The cell membrane is selectively permeable, which means it allows certain things to pass through it and allows other things not to pass through it. So the cell membrane is impermeable, does not let solutes pass through it. But the cell membrane is permeable to water. That's why anytime in any of these solutions that I'm about to teach you, the 3 types, it's always going to be water that's moving in and out. Okay? So for this one, because it's going from high to low, water is going to exit this cell, and this is going to cause this. If this is an animal cell, this is going to cause the animal cell to physically shrink, okay? The animal cell is literally gonna physically shrink, okay? And I'm gonna give you an example of this right now. So, this is also called plasmolysis. We learned that, because plasmo, we know plasma membrane. And lysis, remember hydrolysis. Lysis means breaking down or shrinking. So plasmolysis is when the plasma membrane shrinks, just like how I showed you in this diagram right here. Okay? And then I'm going to give you an example of a hypertonic solution, okay? So we have a fish tank, okay? The fish tank has a little fish in it, okay? This fish is a freshwater fish. So this fish is used to living in freshwater. Because it's used to living in freshwater, it has a low concentration of solute, or in this case, NaCl, which is salt. And then it has a high concentration of H2O, okay? But we place it in salt water. So this water around it has a high concentration of NaCl, or water of salt, and then a low concentration of H2O. Okay? This is an example of a hypertonic solution because the fish has more water than the water around it. What it's going to do is that the water in the fish is going to leave the fish. Okay? So the fish isn't going to die. The fish is just going to feel dehydrated and stuff because it's literally actively losing water. And this is also an example of shrinking and stuff. You know how if you're in the swimming pool for too long, then your fingers start to get all wrinkly? Hypertonic, Tonocity, whatever, okay? The second type is the opposite. It's hypotonic, hypotonic. in this diagram, we have the same cup of water. We have the same cup. And then we have water. But in this one, the water has a high concentration of water, H2O, and a low concentration, I wrote the same thing twice. And it has a low concentration of solutes, okay? But this cell right here has a low concentration of H2O and a high concentration of solute, okay? So, remember how water always moves from high to low? So, this water is going to go from here inside the cell, okay? And so water is going to start entering the cell, and this is going to make this cell swell, S-W-E-L-L, swell, and it might burst. Okay? That's hypotonic. It's kind of like, yeah, you can like search up like examples and stuff. The book doesn't really say a lot, but yeah. I would say read the book for examples if you have time. Okay, the third type is called isotonic. It's when the concentration of solutes is the same as the concentration of water in both the cell and outside the cell. This is basically going to mean that, okay, this cell is going to lose water, but then the outside is also going to give it water. It's going to lose water, give it water. It's always going to stay equal. That's isotonic. It's honestly really simple. You just really have to like memorize this stuff. Is there anything else I missed? Okay. Let's talk. This is like kind of like, this is not extra because this was on our thing. This was on our textbook. This is a unicellular organism. Okay, this is a unicellular organism. So in a unicellular organism, it has like a vacuole, okay? Okay, it has a vacuole. And say, for example, that this is a hypotonic solution. Yeah. Say this is a hypotonic solution and that water is actively entering. These cells cannot afford, and literally write this down, this cell cannot afford to have water enter. Like unicellular organisms can't just let so much water enter. Like they need to control it in a way. Okay? And the way that it's going to control it is that this is called a contracting vacuole. And the way that it's going to control this is that it's going to spend energy pumping out water from out of here to try to like keep it the same. Anyways, that's it.